This embryology highlights screencast concerning nervous system development deals with the peripheral nervous system and meninges. The components of the peripheral nervous system include cranial nerves and spinal nerves, one glial cell type, the lemocyte, also called a Schwann cell. It includes afferent neurons and efferent neurons, where afferent refers to neurons that convey information to the central nervous system, and efferent is away from the central nervous system. It consists of somatic and visceral neurons, somatic carrying information from skin, joints, and skeletal muscle, visceral from vessels and viscera. Two kinds of visceral efferent neurons, which are autonomic neurons, the preganglionic and the postganglionic ones. These postganglionic have their cell bodies in autonomic ganglia, spinal ganglia, and cranial nerve ganglia, where the unipolar afferent neuron cell bodies are located. Somatic and preganglionic efferent cell bodies are in the central nervous system, so only their axons are part of the peripheral nervous system. If we look down below, we can see early developing neural tube. We see a sulcus limitans demarcating the alar plates from the basal plates, the mantle layer and the marginal layer just beginning to show up. So this is central nervous system. On the peripheral nervous system, from neural crest, we see evidence of a spinal ganglion. This would be on a dorsal root, and here would be a ventral root. We see axons that have their cell bodies in the central nervous system, but their axons are part of the peripheral nervous system. The neural crest gives rise to the neurons and glial cells of the peripheral nervous system, and this cartoon shows the different kinds of cells that are derived from the neural crest stem cell based on the type of induction molecules or neurotrophic factors that this stem cell is exposed to. It gives rise to the lemocyte. It gives rise to the bipolar sensory neurons, the peripheral afferent neuron. It gives rise to autonomic postganglionic neurons, the two kinds, the adrenergic and the cholinergic postganglionic neurons. And it gives rise to non-neural tissue, melanocytes, pigmented cells of the skin, adrenal medulla cells that secrete epinephrine, and as you know from embryology, it gives rise to ectomesenchyme, which forms bones and connective tissue and even teeth in the face. Let's look at in more detail at the neural cells that are derived from neural crest. There is just one type of glial cell, a neuralemocyte, also called a lemocyte or a Schwann cell. And that cell migrates throughout the peripheral nervous system. It ensheathes non-myelinated axons. It myelinates larger axons that need to conduct at a more rapid rate. And it forms satellite cells that surround the unipolar cell bodies of primary afferent neurons in cranial nerve and spinal ganglia. It also forms satellite cells in autonomic ganglia. In the peripheral nervous system, all neurons need to be ensheathed or myelinated. This preserves the ionic milieu, so they have the correct portion of ions for conduction. And so all peripheral nerve axons have to be protected by lemocytes. Considering neurons, we have afferent neurons that originate from the neural crest. They originate as bipolar cells. Subsequently, they become unipolar in almost all cases. In some cranial nerves, such as the vestibulocochlear nerve, they remain bipolar. Here we see the cell bodies, the unipolar cell bodies, located in a spinal ganglion, or they could be located in a cranial nerve ganglion. Incidentally, a minority of primary afferent neurons come from placodes, thickenings of the ectoderm, in the case of some cranial nerves, particularly the vestibulocochlear nerve. In the case of efferent neurons, somatic efferent neurons and preganglionic visceral efferent neurons, cell bodies arise from the basal plate of the central nervous system, and just their axons join the peripheral nervous system. And we see that diagram below. Cell bodies are in the central nervous system, but their axons are part of the peripheral nervous system. However, the postganglionic visceral efferent neurons, they arise from the neural crest, and their cell bodies are within autonomic ganglia in the peripheral nervous system. We have paravertebral autonomic ganglia and a sympathetic trunk, prevertebral autonomic ganglia such as the celiac and the craniomesenteric, and we have terminal autonomic ganglia in the walls of viscera, and particularly the gut. Here we see postganglionic visceral efferent neuroblasts migrating away from the neural crest to form the paravertebral ganglia, the prevertebral ganglia, and the terminal ganglia in the gut wall. Also, some of them will migrate to the adrenal medulla and become adrenal medulla cells that secrete epinephrine. Peripheral neurons establish contact with what they are going to innervate very early in development. They make contact with the nearest somite or semitomere or placode or pharyngeal arch, and they never lose that contact. They end up innervating everything that's derived from these embryonic somites, semitomeres, placodes, and pharyngeal arches. Once established, that innervation contact must be retained, even if the neuron has to travel extended distances and make detours to reach a distant target. It has to maintain that contact, otherwise it would lose its source of neurotrophic molecules and degenerate. Perhaps the best example of traveling an extended distance is the recurrent laryngeal nerve. This nerve comes from the brainstem and it innervates the larynx, but it does so via the thorax, because the heart and aortic arch is migrated from the neck into the thorax, causing the nerve to travel down the neck into the thorax and then back up to reach the larynx. In addition to innervating semitomeres, cranial nerves will innervate pharyngeal arches. Each pharyngeal arch is innervated by one cranial nerve, which innervates that arch and all of its derivatives. So, trigeminal nerve innervates the first pharyngeal arch, and that supplies muscles of mastication. The facial nerve innervates the second pharyngeal arch, and that's muscles of facial expression. The glossopharyngeal nerve innervates the third pharyngeal arch, and that supplies mainly one pharyngeal muscle. And the vagus nerve innervates the fourth and sixth pharyngeal arches. The fifth arch disappears, and those arches become skeletal muscles of the pharynx, larynx, and esophagus. Down below we see an early neural tube with the mantle layer very obvious. This is going to form central nervous system, and then we see some evidence of neural crest. So these stem cells are going to give rise to the various cell types of the peripheral nervous system. Finally, let's discuss the development of meninges. Meninges surround the central nervous system and the roots of spinal nerves and cranial nerves. In the adult, there are three meningeal layers: the dura mater, the arachnoid, and the pia mater. The term mater means mother. It arose because ancients thought that the meninges gave rise to the central nervous system. So down below here we see the dura mater. In the case of the cranial cavity, it serves as a periosteum as well as a protective layer for the brain. Deep to that is the arachnoid. That is connected by means of arachnoid trabeculae to pia mater, which coats the surface. In the case of the central nervous system, there's a histological layer of astrocytes that form a glial limiting membrane, and the pia mater is attached to that glial limiting membrane. So these three meninges come from two layers of mesenchyme that surround the developing nervous system. The outer layer becomes dura mater, also referred to as the pachymeninx, because it is a tough, highly collagenous layer. Meanwhile, the inner layer becomes the arachnoid and pia mater, and it becomes that because it undergoes a cavitation that creates two layers out of the one mesenchyme layer. This cavity between the arachnoid and pia mater, which develops by cavitation, becomes the subarachnoid space. The entire subarachnoid space is lined by flattened fibrocytes. This space fills with cerebrospinal fluid, which is produced within brain ventricles and exits to surround the central nervous system and roots within this subarachnoid space. The cerebrospinal fluid within the subarachnoid space provides a buffer that helps to protect the central nervous system. Cavitation is incomplete, and that leaves some trabeculae that run between the arachnoid and the pia mater. Incidentally, the pia mater is intimately attached to the surface, and it dips into every crevice, including perivascular spaces. So the beginning of perivascular spaces are lined by pia mater. The dura mater is mainly collagen, but there's also collagen associated with the arachnoid and the pia mater. Deep to the surface, which is the flattened fibrocytes, there's a small amount of collagen in the pia mater, and there's a delicate amount of collagen within the arachnoid, and delicate collagen within trabeculae. So that concludes this embryology highlight screencast concerning development of the peripheral nervous system and meninges.